welcome to the second Antarctic event series event, Lightning Talks and Social Call for ECRs headed south. I'm Mariama Dryak. I'm the director for the Polar Science Early Career Community Office, or PSECO, as I'm sure you all have heard in some way, shape, or form, i.e. you're here. And we're excited to be putting on this event today that will allow people headed to Antarctica or doing Antarctic science to share with one another and connect. And today we will be working with Casey for technical support. So if you're having trouble with your slides or something else happens, feel free to reach out to Casey to help you. Casey is my colleague at Series Education and Outreach at the University of Colorado Boulder. And at the core of PSECO is inclusion. So we expect that all attendees will adhere to PSECO's community guidelines when interfacing with others on the call. Because we have a small group, we have an awesome opportunity to connect. Let's just do so respectfully. And Casey will post a link to that, and the chat has already. And the format for today is that we'll probably take a second now, actually, because we have a smaller group, to introduce ourselves verbally, and then we'll launch into the lightning talks, slightly adjusted in format from 2-minute presentations to 3-minute presentations if you want that extra minute. And we also have an opportunity to be able to ask folks questions about their research. So in between each of those talks, definitely feel free to unmute yourself and ask a question, or send a question directly to me and I can read it out for the person. And then after the talks, we'll have a little bit of social time to connect more deeply in a non-presentation related format. Finally, if we have an additional slot for these presentations too, so if you want to talk but haven't signed up, directly message me while others are presenting. But before we get started, let's just do an around the room of who's on the call, and that'll allow us to have context for where folks are from, where you're calling in from, and what your research is about in 3 words. We'll stick with the 3-word challenge, which is fun. So my name's Mariama Dryak, pronouns are she, her. I am calling in from Boulder, Colorado with the Polar Science Early Career Community Office. And as I said before, my science in 3 words is Antarctica, glaciers, and satellites. I'm going to popcorn it to Rahul if you want to go again for those who weren't on the call, and then you can popcorn it to someone else.
Okay, thanks. My name is Rahul Dey, and I'm calling in from Goa, India. I work with ice cores, and if you say the 3 words, mine would be Antarctic ice images. Thanks. So over to Casey, maybe.
Over to Casey. My name is Casey Marsh. I work with Mariama at Series Education and Outreach in Boulder. And 3 words, that's tough. I am transitioning I'm going to grad school next fall, but I finished my undergrad at CU, so I don't really have research yet, but I guess I would say community, climate, and ocean for my interests. And I'll throw it to Victoria.
Hi, I'm Victoria Vandersalmen. I'm in Anchorage, Alaska at the University of Alaska, Anchorage. I just started graduate school about 2 weeks ago. And my research in 3 words would be Comasians, adaptations, and Antarctica. And I will pass it on to Nicole.
Hi, everyone. I'm Nicole. I'm a postdoc at Georgia Institute of Technology. I'm originally from Australia, and my research in 3 words is krill swarming behavior. And I will popcorn it to Menzie.
Hi, I am Mansi. I'm from University of Texas, San Antonio. I work with Steve Ackley, and 3 words of my research will be sea ice, Antarctica, and satellite. And I will popcorn it to Gina.
Hi, I'm Gina Joseph. I work with John Cassano at the University of Colorado Boulder. I guess my research in 3 words would be polar, atmosphere, drones. And I will popcorn to Parker.
Hey, my name is Parker Levinson. Pronouns are she, hers. I'm a master's student at Montana State University, and my research in 3 words, Weddell seal demography. And I will popcorn it to Julian.
Hi, I'm Julian Williams. I'm a PhD student at the University of Texas at San Antonio. My research in 3 words, sea ice pictures. Yeah, thank you.
Nice. So to kick it off today, as I sent via email, we'll get started with hearing from Victoria. And I'm really sorry, I don't know how to say that first word of your talk title. So I'm going to let you introduce the whole topic. And yeah, feel free to share your slides now. I'll be keeping folks' time for everyone. And so I'll private message you that you have 1 minute left if you're at the 2-minute mark. And then also indicate when it's 3 minutes. But we have so much time. So there's flexibility. Take it away, Victoria.
Alrighty, I'm just working on sharing my screen here. Oh, it wants me to quit Zoom to be able to share. So yeah, it's not going to work.
Sure, these things are. The settings should be good. Can someone else try to share and see if it's like a local issue for Victoria versus a settings thing?
I can try.
Okay, thanks, Nicole.
Yeah, all good.
Yeah.
So it looks like I have to quit Zoom and reopen to be able to share. Would someone mind going before me, and then I'll just log right back in?
Yeah, Vitor, if you want to kick it off, go for it. Oh wait,
Thank
we didn't
you.
hear from you for introductions also.
Yeah.
Feel free to introduce yourself. I'm sorry we missed you.
I'm Vitor Pavinato. I'm from Brazil. I'm in Worcester, Ohio, at the Ohio State University. 3 words, I think, will be genomics, adaptation, and demography, I guess. So.
Awesome.
Okay.
Do you want to share your screen? And also define the word demography for us. Who are
yeah, I'm going to use from the perspective of population genomics. That's basically estimate the population size through time.
Okay, awesome.
Okay, so let's get started. I'm Vitor Pavinato. I'm population geneticist and postdoc at the Ohio State University. And my research involves the study of evolutionary history of the Antarctica midge with population genomics. My research is part of a large project involving 2 US and 2 UK institutions, and it's funded by the US National Science Foundation and UK's Natural Environmental Research Council. The main goal of this large project is to understand how terrestrial insects adapt to the Antarctica environment using comparative physiology and genomics. And as a geneticist, I'm interested in the comparative genomics among terrestrial Antarctica and subantarctica species and the population genomics of the Antarctica midge, Belgic Antarctica. So the Antarctica midge is the only insect endemic to Antarctica. It lives in patchy island habitat along the Antarctica Peninsula, and it's wingless and shows several physiological adaptations to extreme conditions, such as tolerance to freezing and desiccation. Since this species is originated around 65 million years ago, and since Earth's climate changed a lot, this species, Belgic Antarctica, is an ideal model for understanding the link between an organism adaptation, evolutionary history, to Earth's climate and geological history. As an outcome of this research, it will be possible to predict the species' future persistence due to rapid and current climate changes. I think I'm too fast, maybe. So my specific goal in this project is to sequence. We already sequenced 300 whole genomes. And with these genomes, I'm going to infer the genetic relationship among the populations, infer the demographic history, and search for signature of natural selection that might be associated with climate changes. For example, here I have a plot that shows the demographic population changes through time. That's basically one of the outputs of the demographic inference that we do with genomic data. So it will be my first time in Antarctica, and my goal in this field season next year will be to collect more sample that in never visited island and recollect critical populations based on the preliminary results. Thank you for the opportunity to speak about my project, and I'm happy to take any question.
Thank you. I have a question while we're waiting to see if anyone else does. And if you do have a question, you can do that by unmuting yourself, raising your hand, unmuting yourself and asking it, or again, sending it to me. I'm wondering what specifically you're going to measure down on the ice this summer.
No, I'm not going to do any. Look, we have some logs, temperature logs around some islands that we need to recollect and that were deployed, I think, in 2020, the last time they went there. So, and my goal is to kind of help in the collections. We are going to look for other islands that we don't have samples, and also recollect some other samples from other some population that might be interesting to do like a deeper sequencing with other sequencing technologies.
Awesome. Thanks, Rahul. Do you want to ask?
Yeah, hi. So see, I have literally zero idea about the biology that you were talking about. But then what interests me is these midgets that you're talking. Are they like endemic to that region, or they were transported in like, past some million years or something like that? And
No,
have they evolved?
So they are endemic to the Antarctica Peninsula. They have some close related species that are present in the south of the South America, and also in other northern islands like the Jose Island, something like that. So they have some other related species, but they are endemic to Antarctica. They only live there in the Antarctica Peninsula.
No, so like my question was like, are they like transported from the mainland, or they like developed over there itself?
Yeah, they develop over there.
Okay, thanks.
Thank you. Awesome. Let's transition on to back to Victoria. Do you want to try sharing your slides again? And if it doesn't work, you can send them to me via email, and I'll send you my email.
Okay, sounds great. I think it'll work this time.
Perfect. Beautiful. Okay. Take it away.
Awesome. Alrighty. So my name is Victoria Vandersamen. I'm going to be looking at Comesian biodiversity and adaptations in Antarctica. I'm here at the University of Alaska, Anchorage. My professor is Dr. Sarah Gherkin, who is one of the leading experts in Comesians, and she actually went to Antarctica last September to the Western Peninsula, and so we'll be going to the East Antarctic this March. So again, I just started graduate school about 2 weeks ago. A little bit about me: I'm also a U.S. Coast Guard 100 gross ton master captain. And so I work on a liveaboard in Prince William Sound, Alaska. in the summer. And that's actually how I got started working with Comasians. I met Dr. Gerken as an undergrad, and I started collecting Comasians here in Alaska. So what they are, they're small benthic invertebrates, and they're incredibly important because they feed everything. Fish, birds, even when they're in great enough abundance, they can feed gray whales, which is pretty cool. They're also deposit feeders, so they're able to remobilize carbon from the sediment back into higher trophic levels. So that's their importance. We're using them as a model organism for invertebrate adaptation in Antarctica due to their high diversity and their abundance. I'm going to specifically be looking at a Comasian family called Leuconidae. There are 8 families in the order Comasian. And so what we're going to be doing is using transcriptome analysis to assess the difference in gene expression. And the reason we're doing this is because transcriptomes are actively changing, and it varies on the stage of life that the organism is in, as well as their environment. And so that's what we're going to be doing to look at the adaptations that they may have. What we're then going to do in 2024, we're going to be going to Chile to collect samples. And so we'll be comparing our samples from the Antarctica collections to Chile. And so this is going to be about a 4-year long project. Yeah, sorry, I'm a fast talker. Anyone have any questions?
Yeah, I have one to kick it off. Are they related to krill? Is just as someone who knows nothing about these critters?
I think they are related to krill at the not sure what level they're related at, but I'm pretty sure they would be, yes.
Awesome. Any other questions?
Yeah. What's the goal of comparing the ones from Chile to the ones from Antarctica? Do you want to see like when they moved there, or sort of like the evolutionary history there?
That's a great question. So the reason that we're using Chile and Antarctica is Chile is more temperate compared to Antarctica, which is polar, and they have similar ancestors. And so that's why we're able to use South America to be able to then compare those transcriptomes.
Cool, thank you. And can you speak, I think this might be a consistent question I have, but can you speak to your methods just real quickly in our last?
Yeah, so to collect the Comasians, we're going to be doing a variety of things. We're going to be doing box cores. We're also going to be using epibenthic sleds, Blake trawls, dredge, and then we're going to elutrate the sample and then pour it over a sieve. You want to be really gentle with collecting them because you don't want to degrade any of the DNA, which obviously if you kill them is not going to be very useful. So that's going to be the collecting process, and then we'll store it in RNA later to then process it once we get them back to Alaska.
Thanks, Victoria. Next, we'll hear from what Nicole is saying is a distant relative in the sense of them both being crustaceans. Nicole, do you want to share your slides and share about krill?
Absolutely. There you go. So as I said before, my name's Nicole and I'm a postdoc at the Georgia Institute of Technology. And I'll be looking at Antarctic krill and what makes them swarm. There we go. Fun fact, I'm originally from Australia, so that's why I have an accent. I don't think there's an awful lot of people who go down to bases for other countries. They tend to normally stay within their own country's region. If they're studying Antarctica and the Arctic, they might move at some stage in their career. I moved after I finished my PhD. So I've been south before, but never to a US base. We have lots of data about krill, specifically acoustic data from both the krill fishery and from scientific cruises. So we know the size and the shape and the density of the krill swarms, but we don't know what causes krill to swarm, and we don't know what makes them disperse afterwards, and we don't know what their behaviors are in the swarm. So there's an awful lot of stuff to do with krill swarms that it's just really kind of unknown. So we're running a whole bunch of flume experiments at Palmer Station this season. So we've got both a horizontal and a vertical flume. They're basically just cylinders of water. Horizontal means the flow of the water is horizontal. Vertical means the flow of the water is vertical. It's pretty easy. We also have a concurrent project with a PhD student who's also coming with us. That's a small annular flume. So the water goes around in like a big whirlpool, like a big circle. And we'll be at Palmer Station from October to December. And we're looking at how different stimuli cause them to change their swimming behavior. So we've got photic stimuli, which is light. We've got physical stimuli, which is how fast the water's flowing. And then we've got chemical stimuli. So we've got some chlorophyll, which is a proxy for having food. And then we've got some penguin guano, which is a proxy for having a predator nearby. And we can obviously change all of those and do it as like a big multifactorial kind of experiment. And all of the data that we get from this will then be used to generate an individual based model so that we can look at swarm dynamics, not just under the conditions that we're going to be looking at, but potentially we can then like interpolate and draw out some conclusions looking into the future as well with any sort of luck. So this is like a little sort of test model that we've been building with the data that we've got from a previous season that my PI went down with. I did not join them, unfortunately. But you can see that even just slowly changing one or two sort of variables, we can start to see things that start to look more natural, and we can start to see things that look a bit more unnatural if we start to get them to cluster too close together. Some other fun info about me. I've been south twice, but never to a base. I finished my PhD in 2019, so this is my first postdoc. I actually previously worked with fur seals before I fell in love with krill. I've swum in an active volcano, and at one point in my life, I ate ice cream for 7 weeks straight. Ask me all about it. That's me done.
Nicole, what an awesome note to end on. I have many questions related to the last slide, but first I'm wondering if you can ask or speak to how specifically you use the flumes and in relationship to the krill to make sure what's happening is natural.
Yeah, absolutely. So obviously it's never going to be an exact replica of the open ocean, mainly because they're in a confined space. We're never going to be able to generate the same sort of currents, or we can only mimic as close to sort of the environment as we can, but it gives us an idea of how obviously their behavior changes. So we're only putting 3 or 4 krill into the flumes at a time. The flumes are about sort of 1/2 a meter long, 1/2 a meter wide, 1/2 a meter tall. The vertical flume is slightly taller, and that's just so that we get enough water pressure to actually get like upwelling and downwelling with the pump. But obviously krill are only small. They're about as long as your pinky, that's sort of 6 centimeters long. So having 5 or 6 of them in that space, they rarely sort of interact with each other. So we get to see how an individual krill reacts to those individual behaviors. So obviously with multiple krill, with multiple trials, we can then go, oh, that one just really likes sitting in the corner of the tank, whereas like these other like 4 that were all in the same conditions all, you know, did something completely different. So we can start to work out if it's just them being weird or if it's actually something that we can maybe go, oh, that's something that's definitely like more likely going to be what the population at large is going to be doing as well.
Thank you. Do we have any other questions?
I have a question about what the definition of an actual swarm is in the open ocean. Like how many krill are there in this?
So the definition of a swarm isn't actually necessarily the number. It's all the animals aligning in the same polarity and following the same movement patterns. So if you have a few sort of individuals and they start to come together and they start to loosely follow each other and go roughly in the same direction, that's when it's schooling. And when you get like a big group, but they all very much are sort of systematically, we are this spaced out and we're all turning left at the same time, that's when it starts to become swarming.
Thank you, Nicole. Awesome. And great question, Casey. Rahul, can you share your screen?
Sure, just a second. Okay, so I hope it's visible.
Yes. If you want to do full like presentation, that's awesome. Yeah, great.
Okay, so my name is Rahul. and I am a PhD student, almost at the end stages of wrapping up my work. My work has been involved with an Indo-Norwegian project called Mad Ice, which as part of which what we did is we retrieved 2 ice cores from Zupranen ice rise and Leningrad Kolin ice rise in East Antarctica. It's like over here, right where you see, almost 8-9 degrees on the east, 8-9 degrees east. And the 2 ice cores that we got, we use them to understand the climatic variability over the last 300 years. And using that experience that we gained over there, we are trying to move on to a new project, which is the sea ice and westerly events during the Holocene in coastal Antarctica, which is actually the name of the project, SIWA. So my major tool is visual stratigraphy, or in simpler terms, the images of ice cores, which can be used to find how old the ice is, as well as other physical properties like density, how it is varying, and multiple factors like that, which it's basically one proxy which gives you lots of information. So what is SIWA about? So this project, what we are looking to do is, as part, it's a tri-national project, India, Norway, and UK. And we are trying to get approximately 500-meter long ice core from coastal Antarctica. These ice rises are right to the right of this sites that we have already studied. So we are trying to understand, we are trying to find a record for the last 12,000 years at least. Although we expect much more than that, but then we are trying to cover the last Holocene. And the last season, we had our team of the geophysical team had already gone there. They did some radar soundings, shallow and deep, more than 500 kilometers long of soundings. They did some stake installations, got back some fern cores, 15-meter fern cores, and set up an AWS also. Now, we have 2 ice core sites. So these are candidates. So the geophysical team is basically working on deciding which one should we drill on. And our, the weightage is more towards Camel Riggin here, but then it's still not finalized. And this season, what we plan to do is, this here is Maitri, if you see. This is the Indian Research Station, the oldest sustaining Indian research station. We will be heading on a convoy for approximately 300 to 350 kilometers. So this is the route that we take. The red lines that you see, this is the route that we take over 10, 12 days, I guess. I'm not sure because I haven't done a convoy before this. I have been to Antarctica, but never a convoy. So we are carrying 7 piston bullies with several tons of equipments. Once we get there, we set up camp, and max to max, we drill a 100-meter core so that we have something back, and we can at least, we have 2 candidate sites. One, if we get some data, if it is not good, then we can head on to the next site. So that's it from my side. Thank you.
Thank you. I'll kick it off with a question again. I'm wondering what specific questions are you trying to answer with that 500-meter core?
Okay, so the major question that we want to understand is how the sea ice has been varying, how the westerly winds have been varying, and how the CO2 exchange has been happening in that region. And also, if possible, the deglaciation history. So now that is on the bigger expectations that we have, but then these are the major objectives. And other than that, there is no ice core record from coastal Antarctica, which goes back, or this part of the coastal Antarctica, which goes back to more than 300, 400 years. So this would be a very important, addition to the data sets, how it has been changing.
Awesome, thank you. Any other questions?
I have a question. How do you select your sites that you're going to drill at? Do you use GIS beforehand?
No, so we use GPR surveys, ground penetrating radars, which are basically designed for penetrating ice. So we call them ice penetrating radars. So once the radar surveys are done, we try to understand whether the layerings of these sites are uniform or not, whether the flow, the horizontal flow at the site is minimal, the vertical strain is constant, and whether there is, these sites are basically miniature glaciers. So when you look at them, you can see that these are miniature shaped glaciers. So we try to find out whether these glaciers have moved horizontally or not in any two-dimensional direction. So once we have those information, then we define whether, yeah, this is a possible site. And then these model people, they can always tell you that you go 500 meters, will give you 20,000 years. So that's how we define.
And do you do that in real time?
Technically not, because I am not a geophysical person, but then yeah, the team that does it, they mostly do the surveys and come back here, then do the all the analysis and everything. They don't like working in the field.
I have like a quick question. So your field sample would be in the coastal, you said. So is it like ice shelf or sea ice? Because you mentioned even you're going to study sea ice variations. So how are you going to study sea ice variations?
So we understand the variabilities of sea ice using proxies. So what my study found is that the sea ice variability, like how it is changing with seasons and over time, controls the major variability of stable isotopes that are precipitating into the site. Then there are studies which talk about chemical proxies like sodium, chlorine, which comes from the ocean, methyl sulfonic acids, sulfates. So they are also controlled by the variability of these sea ice changes. So we basically use proxies to understand how the variability has been happening, not the real-time variability actually.
Thank you. Next, we'll be hearing from Gina. Do you want to try to share your screen?
Yes, I will try. Okay, do you see the full slide?
Yes.
Okay, awesome. So I'm kind of presenting on behalf of myself and another student who will be going down to Antarctica with me. And the purpose of our project is to observe the Antarctic atmospheric boundary layer using uncrewed aircraft systems. So yeah, just to quickly introduce myself, I'm a PhD student at CU Boulder, and I'm a 4.5th year. So I'm in the second semester of my 4th year. And Mackenzie is also a 4th year as well. And so we will be deploying to the Seipel Dome Field Camp to fly some drones and observe the atmosphere. So this project, I'm actually only a part of it just to help with the uncrewed aircraft operations. It's not actually for my own research, but my own research has to do with data from the same aircraft in the Central Arctic from the Mosaic expedition. So we have some figures from that field campaign that we're kind of trying to recreate to learn more about the Antarctic atmospheric boundary layer as well. So we will fly what is called the Datahawk 2, which is a fixed-wing uncrewed aircraft system, also known as drone. It's this little aircraft that you can see here. It has about a 1 meter wingspan. It's pretty light, only weighs 1.8 kilograms. Kilograms means anything to you. You can fly it for about 40 minutes. That's if it's warm enough out. If it's really cold, like it might be at Seipel Dome, we might expect more like a 20-minute flight. And it measures temperature, pressure, humidity from a few different sensors. It also measures the IR temperature of the surface and the atmosphere above the drone. It also has a fine wire array for turbulence measurements. So it can get really fine scale temperature and wind. So what a flight pattern kind of looks like from the Datahawk is this circular pattern that goes between the surface and 1,000 meters altitude. So we're measuring all of these things that are listed on the right or on the left with very, very high frequency. The turbulence measurements are at 800 hertz. Some of the other measurements are at 100 or 10 hertz, but it gives really, really high resolution of these different variables between the surface and 1,000 meters. So you can get a great picture of what the atmospheric boundary layer looks like. There's a few different types of flight patterns. I think we'll be mostly focusing on the profiling just up between the surface and 1,000 meters for this campaign. You can also do a loiter at a fixed altitude. So that kind of measures the temporal variability of the same air mass as you're sampling it. If there's any features of interest, it's interesting to do a racetrack flight across a feature of interest. For example, in the Arctic, it's interesting to fly over a lead and kind of see how the atmosphere differs upwind versus downwind. At Seipel Dome, where it's just ice everywhere, maybe we won't really be doing these too much. But yeah, this is an example figure from the Arctic that kind of just shows what a temperature profile might look like. These are all the temperature profiles we collected during Mosaic. But it's, yeah, you can kind of see how high resolution these are. These are color coded by time as well. That's why they are in different colors. So yeah, the goal is to just collect these measurements at Cycle Dome, where there's not a ton of very high resolution measurements or measurements at all of the atmospheric boundary layer, and then somebody else will analyze the data once you return to figure out what we have learned. And that's it.
Thank you. How do you make sure that, let's say, really crazy katabatic winds aren't affecting the stability of the aircraft and what it's measuring, or is it built specifically to be stable?
So the aircraft can operate in not too high of winds. So the general criteria we fly, at least this is what we did on Mosaic, which I imagine will be similar in Antarctica, is surface winds about 10 meters per second at the most. So we're not going to be, if there's like a really high wind event, this aircraft can't handle it, but it can handle a decent amount, especially, yeah, if you encounter high winds aloft and the aircraft can't keep up, you just kind of bring it down. But it's pretty stable in terms of its ability to stay, you know, at least level, and you can just bring it down. And most of the flight occurs in autopilot. So yeah, there's systems built into the aircraft that kind of help stabilize it.
Awesome. Any other questions?
I have a quick question. Do you have to take multiple of these little drones down with you on the off chance that something does happen and they break or a sensor goes wrong?
Yeah, we're bringing 4 of the drones with us, and we're expecting to operate for about 2 weeks. So that's kind of a decent amount of backup. In addition to the 4 drones, we have a ton of replacement sensors, motors, etc. So yeah, there's expected things to go wrong and have to replace some stuff.
That's awesome. Thank you so much, Dina. And next we'll be hearing from Julian.
Hi.
Hello.
Can you see the screen?
Yes.
Okay. Hi, I'm Julian Williams. I'm a PhD student at the University of Texas at San Antonio. So I also work with Mansi that's in the group. And today I'm going to talk about sea ice flooding in the southern oceans. I'm going to focus mostly on the Amundsen Sea, since that's where we're focusing on in this study. But we do look at the West Weddell Sea as a comparison to that. So ice flow flooding is something that is a phenomenon that we see in the southern oceans, where the weight of snow on top of the ice becomes too great for the ice to continue at its current freeboard level. So the snow pushes the ice down, and then you get that slush layer where the water is able to encroach on the surface. From there, you'll have a situation where when the ocean water is warm enough, you'll have bottom melt while there's that slush layer still on top of the ice. And when the conditions are cool enough again, so you have now the receded bottom melt of the ice, while the previously flooded slush layer has now cooled off to become what is considered snow ice. So if you think about maybe what it looks like when you put cotton candy in water and just continuously put more cotton candy on top. So you have like this net zero change in the total ice thickness. And this is an indirect input of the sea ice mass balance. So this is another freshwater input into the system. So what we're doing is looking at SAR images, and SAR is notoriously ambiguous. You will see because in in the images, you'll be able to tell where these flooded ice flows are, but it's not as clear as it would be, say, for example, in an optical image. So what we're trying to do is to find and quantify these areas that should be flooded. So in this scene that's in the Amundsen, we're looking at the same scene. It's the same geographical area, but we do think it's best to look at a bit of ice with respect to a given iceberg. But if we're to look at the same place, we'll be able to see what the mean backscatter intensity is. And we should see a change in the backscatter intensity with a change in the conditions of the ice surface. So we see at the beginning or just going into the winter period in Antarctic, how we have a more negative change in the backscatter intensity. So we see a greater or more negative backscatter intensity going into the winter period. So we do think that we should be able to see flooded ice flows. It's been reported in the field. It's been reported with buoy data as well. So we should be able to quantify this in SAR and see how this changes from the summer to the winter period in both seas, the Amundsen and the Western Weddell Sea. Thanks.
Thank you, Julian. Can you break down SAR for folks who aren't really familiar with remote sensing?
Sure. SAR is synthetic aperture radar data. So it's Sentinel-1 in particular that we're using is a C-band frequency. So it's not like optical images where you have multiple bands, or like you'll see like a picture of something and it has red, green, blue. In this case, we have two polarizations, horizontal, and horizontal, vertical. So it's right looking as well. So it's always being acquired at an angle. So there are different band math and image processing techniques that we have to go through with SAR to be able to tell the same sort of features that you would more easily see in an optical image.
And what's the benefit of using it?
Well, with SAR, that's the great thing about using SAR, even though it tends to be a little bit ambiguous by itself, is that you can have looks during polar nighttime when it's overcast and it's dark and you can't actually see anything. So it's not inhibited by clouds or any of those conditions. But optical images are, so.
Yeah, thank you. Any other questions?
Is that it?
Okay, thank you, Julian.
Thank you.
And finally, we have a presentation from Mansi. If you want to share the screen.
So we already did our introduction, but just hi, I'm Mansi. I work with Julian at University of Texas, San Antonio. I'm a third-year PhD student, and my research is funded by NASA's Finest Award, and I work with Steve Ackley. So my research interests, it basically involves studying ISAT-2 data and exploring its capabilities. So here you can see is the ISAT-2. So ISAT-2 is an altimeter data. So it gives data sets in the form of point clouds. And we use that to quantify whatever we need. And there are like range of products from sea ice to glacier, land ice. And what I have been using is one of the primary products, which is a rough raw data. And I calculate the depth of the melt ponds in Antarctica. And the other one is to use the sea ice freeboard product to understand the thickness variations in the Weddell Sea. So I'll just talk a bit about the sea ice thickness variation. So it's just ISAT-2. This is how the freeboard gives. So it is any ice above the water level is what is measured by the ISAT-2. And what we do is calculate the thickness from this total freeboard product. And what we particularly have decided to go ahead is to use empirical equations instead of field measurements or any other satellite data to have less error. And because this is like a new satellite, comparatively newer satellite data, we did not have the field snow depth data around that time. So that's why we decided to go ahead with empirical equations. And we'll be using this to basically understand how ISAT-2 gives the thickness, sea ice thickness variations. And later on, I'll be also using modeling one-dimensional ice model to compare the ISAT-2 results with the modeling results and see how far or how good the products are. We're also looking at retrieving some of the field data sets, if possible, around this time, whenever there are icebreakers or cruises going. If we get that, that would be a great validation of our data set. Other than that, I mentioned about my other project, which was on the melt ponds. So this is mostly to basically detect if there is a melt pond in the ice shelf region. Right now, I've just done a basic melt depth detection, the depth of the lakes. The idea is to understand how ISAT2's return time can help us understand whether there are lakes or not in a specific area. And yeah, that's it.
Thank you, Mansi. I'm wondering how your learning curve with ISAT2 has been, given that it's so new.
It's been, it took a lot of time. But there are this ISAT2 hack weeks, which are uploaded online, which is a great resource. And then I also work with one of the person who is already in the ISAT2 team from the NASA Goddard. She has helped a lot to understand how the data sets is. And I hopefully in future, if anyone uses it, I would be able to do the same.
Yeah, it's great that there are a lot of open source things that are happening where people are sharing the learning curve process. Yeah. Any other questions?
I can ask one. When you said that you have two different research interests, kind of, is that what you were mentioning? How do those overlap at all, or how are those connected with what you're doing?
No, they do not overlap, or they're not connected, because both of it basically started when I started my PhD. It was more like exploring, and I started with the melt pond and that research. And then because my university, Steve Ackley is like a person who is a sea ice scientist, and then I kind of diverted with his research interest.
I see.
Awesome. Any other questions? Thank you, Mansi. Thank you, everybody. We're wrapping this up a half hour earlier just because we had less presentations, but we fully encourage you to share your information if you feel comfortable doing that. And Casey will post a link to where you can do that in the same Excel sheet that was for presentations. We've added a little bit where for folks who didn't present, you can put your details there too, and you can connect with one another later offline of this call. And we hope that you learned a little something new from the lightning talks today. I think it's so inspiring to hear about what all the different niches of and disciplines are doing in terms of really rad cutting edge science. And I want to encourage everyone to go to the Antarctic governance talk that will be held shortly. There will be a link posted to that in the chat too. And also, in case it's something you've thought about or not yet heard of, but still want to apply to, you can apply to the Belonging, Accessibility, Justice, Equity, Diversity, and Inclusion workshop that PASECO is running, and that begins next week. And the content is incredible. And so if you want to be a part of helping make this inclusive environment in polar science wherever you are, I think that'll be a good first step towards doing that. And finally, I hope you all have a really great day, and thank you for coming and sharing about your science and connecting. And we hope to see you soon.